Hello to our friends joining us via recording. Today we are going to finish up lesson number 6. Then we're also going to move into lesson number 7. So before we dive into lesson number 7 about the skin with our newly decorated Integumentary System classroom, we need to do a little bit of review from lesson number 6, which was all about the Central Dogma. For my friends who are here in the chat, can you remind me, the Central Dogma sounds really scary, but it's actually not that scary. What happens in the Central Dogma, or what's the goal of the Central Dogma? The stuff that was lesson number 6. What's our goal? Yeah, I like it. Audrey outlined it really simply. The Central Dogma is the way that we go from the DNA that we store in our cell that has all of our information into RNA that we can use as specific directions for the kind of protein we want to make. We turn that into a protein, and that's the Central Dogma. So the Central Dogma, in the simplest possible terms, is the way we turn DNA into proteins. For us to turn DNA into proteins, we have to start with DNA and turn it into something called RNA. So let's work through this table together to make sure we remember the differences between DNA, which is what we start with in the Central Dogma, and RNA, which is what's in the middle of the Central Dogma. What makes these two molecules different right off the bat is the sugar that they have inside of them. This is why these two molecules have different names, because they have different sugars in them. So let's start over here with RNA, because the R is a shorter sugar. What does the R in RNA stand for? The R in RNA. Yeah, exactly. R stands for ribose. The ribose is the kind of sugar that's in RNA. If we technically wrote out its whole name, it's ribonucleic acid. Ribonucleic acid, that's RNA. So RNA, the sugar that it has in it is how it got its name. It's called ribose sugar. When we talk about DNA, the sugar that DNA has is something called deoxyribose. Deoxyribose. And it sounds scary, but literally all it means is I stole an oxygen from ribose sugar. Everything else is the same as ribose sugar, except I'm missing one oxygen. But that one missing oxygen is enough to make a really big difference in the stability of the molecule that we're making and the kinds of nucleotides that it can have. So let's start with DNA. If we are missing that oxygen, what are the 4 nucleotides that I can use? What are the 4 nucleotides in DNA? Yeah, Kira outlined them for us. Very easy to remember. A, T, G, and C. Those are my 4 nucleotides in DNA. By the way, which one is Dr. Ollis's favorite? Do you remember? My favorite nucleotide, which is what I named my cat. Yep, A for adenine, my Addy cat. So A is Dr. Ollis's favorite. A is found in DNA and in RNA. What are the other 2 bases that are found in DNA and RNA? Besides adenine, who else is also found in RNA? Yeah, C and G are also found in RNA, so DNA and RNA. But remember, the other one that RNA has, because it can't have thymine, is it has uracil. So the 4 bases in RNA are A, U, G, and C, whereas in DNA, the 4 bases are A, T, G, and C. Yeah, so I'm seeing a couple of notes of ways to remember the bases. I like that I think a couple of us have mentioned there's the idea that the car goes in the garage to help you know who pairs with who. We're going to do complementary DNA and RNA in a moment. C and G always go together like the car goes in the garage. And then A and T go together as well, like apples in a tree. That's perfect. So A and T go together, G and C go together in DNA. When I'm doing RNA, remember that T, thymine, is too cool to hang out with ribose sugar. So there's no thymine when we're using ribose sugar. RNA doesn't have thymine. RNA has uracil instead. When we talk about DNA versus RNA, which one of these is single-stranded? Which one is single-stranded? Yeah, RNA is single-stranded. And by the way, if you noticed on this week's bulletin board, there was a little joke about that. DNA is chatting with RNA and says, still single, and RNA says, of course, always. So little joke about DNA and RNA there for you. DNA is the one that's double-stranded. DNA is double-stranded. And again, a lot of that comes down to the fact that we have deoxyribose sugar in its backbone. So it's in a shape where it's easier for it to be double-stranded. But as someone also mentioned, if you're single-stranded, you have the ability to leave the nucleus. So when we talk about RNA, one of the forms of RNA that we talked about is mRNA, M standing for messenger. So messenger RNA is like the chat says, the lucky one that gets to leave the nucleus. So leaves the nucleus for the cytosol. That's the main kind of RNA that we're focusing on this semester, but we did also mention the two other kinds of RNA that are tRNA and rRNA. rRNA, that R stands for ribosomal RNA, and for tRNA, that T technically stands for transfer, but remember, I like to say it kind of stands for translator, if you will. So the two other kinds of RNA, all of them have ribose sugar in them. When we talk about DNA, where is DNA found, by the way? Why do I have to make RNA? It's based on the location where DNA lives. Yeah, DNA lives inside the nucleus, always, 100% of the time. The only time that DNA is not in the nucleus is when the cell is dividing. And the reason that the DNA is not in the nucleus is because there is no nucleus. The nucleus goes away when a cell's dividing. So DNA is always inside the nucleus. That's why I have to make RNA for it to be able to leave the nucleus. This is a table that you have in your notes, and I'm telling you now, it is an underline highlight star table. We want to make sure that we know the differences between DNA and RNA. That's a really important compare and contrast to make. We also want to make sure that we know how to build DNA and how to build RNA. Again, this is something that I will ask you to do on the exam. So when it comes time for exam 2, you will have to do just a little bit of typing in complementary sequences. So let's start with reminding ourselves what this word complementary means. If we are a complementary sequence, we are or are not identical. If we're complementary, we are or are not identical. Yeah, we're not identical. It's not the same. Remember that complementary is kind of like a mirror image. So what I mean by that is I can use what I see in my DNA sequence to predict what I would see in the other sequence, or to predict what I would see in the RNA sequence. So when we're looking here at this DNA sequence, anytime I see a T in one DNA strand, what would I see in the complementary strand? What would go underneath it if I've got a T? Yeah, T, we're always going to pair with an A. T always pairs with A. So every time No, I skipped one, but every time we've got a T, we're always going to put an A. Yeah, Audrey's totally right. My favorite. When I have an A in the original DNA sequence, what am I going to have in the other one then? If T goes with A, yeah, A goes with T. So they're always partnered with each other. A goes with T. So we can do that all through the sequence. And when we'd have a C in the original DNA sequence, we're going to have a G in my complementary sequence. So anytime I've got a C, I'm going to have a G. And every time I've got a G, I'm going to have a C. So we could go through and write the whole sequence. I know that looks absolutely terrible. Y'all were better with typing it. I should have typed. But here's how we build a complementary DNA sequence. So if the question asks you when you're doing the homework or when you're doing the exam, if the question asks you for complementary DNA, that's really easy, really straightforward. It's always just A's and T's, A's and T's, C's and G's, C's and G's. If I tell you to build me a complementary RNA sequence, that's when things get a little bit more funky. When I ask you to build me a complementary RNA sequence, remind me again, which nucleotide can I not use if I'm doing RNA? Who's too cool for RNA? Yeah, T is too cool for RNA. So anytime that I would have normally put a T into my complementary DNA, now I'm going to just sub that out for a U. So if we were building complementary RNA to this DNA sequence here, we'd still have our A. A is still the same. Let me get my pencil back. A is still the same because T and A still go together. But now I have an A in my original sequence. I can't put a T in there. I'm going to have to put a U. So A goes with U. If I have if I have an A in my original sequence, I'm going to put in a U. If I have a T, I'm going to put in that A. Notice my next one was G. G is just fine. C's are just fine. I hit another A. I'm all good until I hit my next T. Then I have to trade out that T for a U. So part of why I had you build both DNA and RNA is to show you that complementary DNA and complementary RNA, they're very, very close to each other. The only difference is that U. And remember, we talked about how if you see a question on the homework assignment or on the exam, any sequences that have a U in them, you know that you're looking at RNA because DNA never has U's. And any sequences that have T's, you know you're looking at DNA because RNA would never have a T. So look for those T's and look for those U's to help you out with figuring out what kind of genetic information that we're looking at. I know one of my groups actually took it a step further. So let me pull up my slideshow here from lesson number 6. One of my groups went as far as starting to do translation on our sequence. So translation, remember, is when we have to use this funky table. So when we do translation, we use those sets of 3. So remember that ribosomes that help us do translation, they're still learning to read. They can only do those little things called codons. They can only do 3 letters at a time. So ribosomes look at a codon. For example, make something up here and hope that I don't mess up. Ribosomes see a sequence that looks like this. They're going to read it in 3-letter chunks. So the first 3-letter chunk they see is A, U, G. So my ribosome, so if we're pretending we're ribosomes, we're trying to read this. We go over to our table. Our first letter tells us which of these 4 rows that will be in. So this is letter A. We're going to be in this row. In this row then, I'm looking for the column that lines up with U. So my second letter tells me which column I'm in. I know I'm going to be in this column. So we had this row for A. We had this column for U. And then I can find my G right here. Or if you don't want to look inside the box, you can go over here to the third letter on the right side. So my codon, my short word that I'm reading is AUG. That tells me that the amino acid I'm looking for right here is methionine, or MET. That's its abbreviation. Okay, so we've read our first codon, AUG. Now we're going to go on to our second codon, CCU. So I go back to my table. My first letter is a C, so I'm going to be in this row, first letter C. Now I go to the top, and it's another C. I know I'm going to be in this column. So C, C, and U. My last letter was a U. That tells me that I'm going to do proline. That's the amino acid that I'm looking for. Now, remember when we were talking about this table, we can see it really well here with proline. Remember that this table shows us that the genetic code is degenerate. Degenerate. What that means is that more than one sequence gets the right amino acid sometimes. So more than one amino acid sequence, or more than one RNA sequence, can get me to the right amino acid sometimes. Remember that the error, or the place where I can change my sequence, is only that last base. So see how here all of these give me proline because I only changed my third letter. If I changed my second letter, that's going to lead to leucine. If I change my first letter, that's going to lead to something completely different. Remember that the genetic code is degenerate, meaning I can have some errors, but I still might get the right amino acid. Does anyone happen to remember, by the way, I have a special name for the nucleotide that I can change in these things? What's the name of that third nucleotide in a codon that I sometimes have the ability to change a little bit and I might still get the right thing? Exactly. Yep. So what that third nucleotide is called, the third nucleotide is called the wobble base. Here you go. Y'all wanted me to sing? Here it comes. It's the wobble base. Yep, as in wobble baby, wobble baby, wobble baby, wobble. There you go. You got Dr. Ellis to sing. That's as close as we're coming right now. So that wobble base, I can change that third one. I can switch it out. Maybe it was supposed to be CCU, but RNA polymerase mixed up and put in an A instead. Or RNA polymerase mixed up and put in a C instead. We're still okay as long as the only thing that changes is the wobble base. So the degenerate code, I can have a little bit of wobble room, if you will, instead of a little bit of wiggle room, right? I can have a little bit of wobble room as long as it's that last base. The last one can change. I saw a couple of questions that I want to address. One of those questions was about whether we need to know the actual names of the amino acids or just the abbreviations. I'm just going to make you know the abbreviations. I think you should have a practice, you should have this on the homework assignment where I ask you to actually do some translation. And you'll see that when I ask you to do that, there's a drop-down menu that includes all of these abbreviations. So you don't need to know their actual names. You just need to be able to use this table to match it up with their abbreviations. I'm not going to ask you to know their full names. And I think a couple of people were helping with this question too. The question about the stop codons. So the stop codons are labeled here in bold, just like my start codon is labeled here in red. When I get to a stop codon in my RNA sequence, what happens at a stop codon? What's going on at a stop codon? Yeah, so Kira gave us a good detailed description for it. When we get to a stop codon, translation stops. It's the end of the road. So we get to this codon. Remember, I told you jokingly that ribosomes, they're calling out to the environment these sequences, and then the tRNA comes over and says, I can tell you what that means. You're good. When we get to a stop codon, the ribosome says, UAA. Can anyone tell me what that means? And no tRNAs come over to help. And so the ribosome says it again, UAA. Who knows what this means? Nobody responds. Nobody's there to help. So then we get to the yelling, like Kaylin said, UAA, come on, somebody tell me what this means. If nobody can tell the ribosome what it means, the ribosome literally falls to pieces. It falls apart. And that's the end or the stop of translation. So proteins end at stop codons. That's the end of the line. Even if there's still nucleotides that are left over. So here, let's go back to our strand that we made. So I'm going to add a stop codon in here, and then I'm going to add another couple codons after it. Here's the thing. If my ribosome is going through, divide up my codons here, and it's reading this sequence, we already did translation on these two codons. These code for something. When I get to UGA, my ribosome reads UGA, and that's a stop codon. This is the end of the line. So any of these codons that come after it, the ribosome is never going to read them. That's it. This is an idea that's important for you to think about when we're thinking about those wobble bases, or when we're thinking about potential mutations in DNA or in RNA. If we change the sequence of what should be there to the sequence of a stop codon, so if we accidentally mix things up and we put a stop codon in there, that's the end of the road. Like, you're no longer listening. The ribosome can't do anything else. So if I put a stop codon right here, when that stop codon was supposed to be 20 amino acids later, that's bad news for my protein, because it's not going to get all of the information it's supposed to get. So stop codons are great when they're in the right place, when we're at the end of the protein. If we happen to accidentally put one in the middle of a protein, bummer for us, because that protein's not going to work. I like what Kaylin said in the chat. She said it's the importance of good communication. Yeah, no, it's 100% true. Like, if I've told you all, my husband is a therapist. So if you are not fighting fair, as your therapist would say you need to, if you're not fighting fair and you throw in some really nasty insult in the middle of what you're trying to say, man, they're not listening anymore. You're not going to get that important stuff in there. So just like we have to communicate correctly, we need to not say things that make people stop listening, right? Stop codons, exactly the same way. Don't toss those around, not until you're ready to be done with the protein. So translation ends when we hit one of these stop codons. Once we hit the stop codon and we're done, then my protein can be released. We're going to go fold it up and make it do its job. Yeah, Kaylin extended our metaphor even further. If you have a bad fight and you're not communicating well, your relationship will fall apart too. Yeah, this is very true. You know what? After class today, legit, I'm gonna talk with my husband about this, and he's gonna really get a kick out of it. So, thank you, Kaylin, for this new topic of nerd conversation for me to have with my husband. Before we move on to talk about what can go wrong in this process of transcription and translation, are there any last minute questions about this or about that group work that we did at the beginning of class? Awesome. Okay, well, I'm seeing a lot of comments that were good. So I'll go ahead and move on. We're going to look at now with the disease sickle cell anemia. We're going to look at what happens if I change something that's not the wobble base. So if I change the wobble base, there's still a chance that I'll be fine. We may never notice that, and it's not a big deal. If I change something different from the wobble base, now I can start getting into some issues, especially when it comes time for translation. So let me pull up right here. In the disease sickle cell anemia, here's something that should maybe be slightly frightening for you. The disease sickle cell anemia, with all of the problems that come from it, all of those problems, whether it's dying tissues or extreme pain or all that kind of stuff, that all comes down to a one nucleotide mutation. As in, my DNA just has one letter that's wrong, and suddenly all of my red blood cells change their shape. It's crazy. So sickle cell anemia, we start with a problem here in our DNA. I've got a codon, or a set of 3 letters, that's normally supposed to say GAG. That's the normal codon. In patients who have sickle cell anemia, instead, that codon says GTG. Now, if my DNA says GTG, or I guess actually technically the one we're reading right here is the CAC. If this is what my DNA says, when I go to make my RNA, I'm going to make the codon GUG. This is what my RNA is going to read because we're complementary to our strand down here. What I'm supposed to make instead of GUG is I'm supposed to be making a GAG. So complementary to CTC is very different than complementary to CAC. When I use my genetic code table to translate these two codons, I end up with an amino acid called glutamic acid in normal situations versus valine when I have sickle cell anemia. If you recall from our discussion of proteins from lesson number 5, when we talked about our amino acids like glutamine or glutamic acid and valine, remember that our amino acids had different personality types. So some of my amino acids have the personality type that's nonpolar. Some of my amino acids have a personality type that's polar, or that's positive, or that's negative. If we change on our genetic code table, if we change that wobble base, we might still end up with the exact same amino acid. That's the best case scenario. The next best case scenario is maybe I put in a different amino acid, but at least it has the same personality type. At least it's still nonpolar. At least it's still polar, or still negative, or still positive. Where we get into trouble in sickle cell anemia is my normal amino acid is down here, it's glutamine. This is normally what I would put into my protein if I have the right codon. When I have that mutation, instead of putting in glutamine, I'm going to put in valine. I went from something that's polar to something that's nonpolar. Remind me, because it's been a little while, when I say that valine is nonpolar, what does that mean about it? How does it feel about water if it's nonpolar? Yeah, when we're nonpolar, we are hydrophobic. We are saying no thank you to the water, don't want to be next to water. What about if I'm polar? How do I feel about water if I'm polar? Yeah, polar loves water. Polar is hydrophilic. So we go from normally having an amino acid in my protein that loves water, that is a huge fan, to an amino acid that can't stand it. It can't even. So here's what happens. When we have our normal chain, here's my normal chain here. When I have a mutation in my codon, it switches out my 6th amino acid in this chain. So at the level of my primary structure, at the level of the sequence of my amino acids, I traded out one of my amino acids. When I trade out that one amino acid in my primary structure, my secondary structures and my tertiary structures change as well. So we go from this heart-shaped chain protein to this, I kind of like to call it an elf shoe. So here's the foot going into the elf shoe here. Remind me, by the way, we can't really see secondary structure in this picture because I kind of made it all a glob. There are 2 options for what the secondary structure is of a protein. What are those 2 options for what secondary structure is? Yeah, so several of us are mentioning, one of them is called an alpha helix. That's that curly Q ribbon. And the other one is called, a beta helix. Yeah, I'm done. The other one is called a beta sheet. That's what I get for reading and writing at the same time. Alpha helix and beta sheet. If I trade out the valine here for the glutamate, if I trade those out, I'm going to mess up the patterns of my curly Q ribbons and my accordions. They're not going to be right. And if they're not right, my overall 3D shape, that's my tertiary structure, that's not going to be right either. And if my tertiary structure is not correct, my quaternary structure will be wrong as well. Can you remind me, what is the quaternary structure of a protein? What is the quaternary structure of a protein? Yeah, so quaternary structure is when I put more than one tertiary structure together. So I've got more than one amino acid chain that's folded up, that's got a 3D shape. When I put more than one of those chains together to build something that's even bigger, made of multiple parts, that's what I call the quaternary structure. Now, when we're talking about hemoglobin, hemoglobin normally exists inside your red blood cells in these nice little sphere-looking structures here. So the normal hemoglobin protein is actually made of 4 different chains, and we looked at this actually in our protein structure lecture. So we have 2 amino acid chains that are called alpha chains and 2 amino acid chains that are called beta chains. Hey, I need you to do me a favor and I need you to write something down. These are, well, here, let's go up here. See how this is labeled the beta subunit? Please, please write down for yourself that the beta subunit is not the same thing as a beta sheet. Please write down that this is not the same as a beta sheet. The beta subunit is 1 amino acid chain. I know it's confusing that they use this alpha and beta thing, but this is 1 whole amino acid chain that's folded into a 3D shape. I call it the beta subunit that I folded into this 3D shape here. For you to build a hemoglobin protein, we take some 3D proteins that are called beta subunits, and we attach them to some 3D proteins that are called alpha subunits. Again, the alpha subunit of the hemoglobin protein, this is different from an alpha helix. This is going to be something that has alpha helixes inside of it, but this is not an example of secondary structure. This is something with tertiary structure, a 3D shape. So that's why when we're talking about hemoglobin right here, we can say that it has quaternary structure, because each of these 4 pieces here, each of these pieces have a 3D shape. Each of them have tertiary structure. So when all 4 of my amino acid chains in hemoglobin are the right shape, the right 3D shape, this is what hemoglobin looks like. It's little individual proteins that fill up your red blood cells completely. Red blood cells, fun fact about them, they're actually so good at transporting oxygen that they don't have a nucleus anymore. They're completely filled with these hemoglobin proteins. They push out their nucleus. There's so much hemoglobin. So normally, hemoglobin fills the entire cell. I find it in these little detached pieces. But check this out. When you have sickle cell anemia, and we've traded out our amino acid for something that is non-polar, when I try to fold my beta subunit chain into the right shape, I can't do it. I end up making a space that's exposed, that's open to the environment, that is hydrophobic. Hydrophobic. Remind me again in the chat, why is hydrophobic bad for a protein? Why, or what does hydrophobic mean? Let's put it more plainly. What does hydrophobic mean? Yeah, exactly. I'm afraid of water, right? I don't like water. If I'm a protein and I have a hydrophobic region, that is a part of my protein that can't touch water. So I put these beta subunits together with the alpha subunits, and they've got these parts that are sticking out here that are hydrophobic. They can't even with water. So instead of floating around and being happy by themselves like normal hemoglobin proteins, these sickle cell hemoglobin proteins end up actually attaching to each other, because you've got a hydrophobic region here and I've got one here. And if we squeeze together, remember those things that we talked about called hydrophobic attractions? Hydrophobic attractions. You hate water, I hate water. Let's squeeze together and make sure there's no water. So I build these proteins that have a region that don't like water, and those regions end up attaching to each other, and we build these big long proteins like you can see right here. These big long proteins look very different than these little individual proteins that we see over here. So the little individual proteins that we see over here is what hemoglobin is supposed to look like. This is what hemoglobin looks like in a sickled red blood cell. Hey, remind me, way back on day 1, we talked about anatomy and physiology. Anatomy and physiology, related or not related? Anatomy and physiology. Yeah, they're absolutely related, right? They, as Tien said, they dictate each other. We have to have one to be able to have the other. So if I have this anatomy, this big long line, compared to this anatomy, these little floating around proteins, not a trick question. Am I still going to have the right physiology if I look like this? Can I still do my job if this is my appearance? Yeah, absolutely not. Several of us are saying that. No, we can't. And that's the deal when you look at red blood cells that have this clumped hemoglobin where it's all stuck together. Not only does it make it harder for these proteins to attach to oxygen, but it also makes it harder for the red blood cells to travel throughout your body. It's already not easy to be a red blood cell. Our red blood cells in the body are constantly having to squeeze into those little tiny capillaries, the really small red blood cells. We're constantly breaking down worn out red blood cells because they get so worn out moving through the body. Well, if you make your red blood cell into this shape, because those hemoglobin proteins stretch it out, kind of like tent poles, if I get all stretched out like this, you better believe that I'm not going to fit into those tiny capillaries. Just like Temiteo said, absolutely, we're going to clog up those blood vessels. And when we clog up the blood vessels, any other red blood cells that would have been able to fit into that place, they can't get there anymore. And if I can't get red blood cells to places, I can't get them the oxygen that they need. So sickle cell anemia is a disease that causes an incredible amount of pain. That pain is actually coming from literally cells dying because they can't get the oxygen that they need. There's some very interesting information, by the way, or very interesting current events in anatomy articles out there. They're trying, if any of you have heard of the technology called CRISPR, where we actually change the DNA in a person's cell. They're actually doing that for patients with sickle cell anemia. They're a very small cohort of people they're trying this with, where they're changing their DNA to get them back to our normal sequence. So let me go back here. Going in and modifying the DNA so that when the body makes new red blood cells, instead of making the mutant protein, they're actually making the normal protein. So if you're more interested, if you want to learn more about sickle cell and how we're using science and technology to treat it, there are some articles about that in current events in anatomy. I'll mention one other thing too. You've probably heard on the news about the coronavirus variants, right? So we're hearing about variants like the South Africa variant and the variant that's over in England. When we talk about variants in the virus, what we're talking about is the virus doing the same kind of thing like we see happening here. So when the virus replicates its genetic information, if it changes some of those bases when it's doing replication, what could happen is that's going to change the codon that we use to build the proteins that are on the outside of the viruses. It's not a big deal if we change the wobble base, right? Just like in our cells, if we change our wobble bases, we still might end up with the same protein. From the human perspective here, if the coronavirus changes the sequence of its spike proteins and we still get a protein that's exactly the same, vaccines are awesome. We can fight it, we can treat it. Where we get into trouble, just like with sickle cell, is if my virus changes the sequence of its amino acids to where my spike protein looks really different from what the vaccine is looking for, it might be able to evade the immune system. So on the news, next time you hear them talking about the variants, what we're actually talking about is our virus has replicated so many times in so many different people, it's gotten a little bit sloppy, and some of its amino acids and its proteins have changed. If we get enough change, it might be able to evade your immune system. So now you know a little bit more about the physiology of how these variants work with coronavirus and with your own cells and stuff in general. Before we leave behind the central dogma, do we have any last minute questions that we want to discuss from lesson number 6, the central dogma? Or I need you all to shoot me an emoji. I'm not feeling the love. I need some emojis here. Here comes my penguin and my dance party. Oh, Fanchon sent me the love. That's excellent. Audrey sent me a penguin too. I love it, Kira. A gold star. Thank you for that gold star. I worked hard to earn that star, man. Oh, more love from Shanti. Thank you. Perfect. I'm feeling more of the love. That's excellent. Timoteo, I am not familiar with what you mean by the AS genotype. Can you clarify that for me?
Hello, can you hear me?
Yes, I can.
Okay, so with the AS genotype, they typically say because they have the S trait of the sickle cell, but then they have the A normal gene. So they call them like they have the trait of sickle cell, like they carriers.
Got it. Yes. Okay. Yeah. So I know what you mean. Yeah. So for someone like you mentioned, they call them carriers. So that's totally right. What it means is that half one of their chromosomes has the normal DNA sequence on it, and the other chromosome has the sickle cell trait on it. So it has a mutation for it. So in those individuals, they and actually all of us, we have two sets of every gene. We just normally turn off one of those sets of the gene. So for individuals that have one copy of the mutated gene and one normal copy, a lot of times we would say that they have the sickle cell trait, which means that a lot of their red blood cells will be the correct shape. Some of them will not be the correct shape. When they have children, they will either give their children the mutated DNA or the normal DNA, and depending on whether their partner has the mutated DNA or the normal DNA, they may have a child that has full-fledged sickle cell anemia. But yeah, if you are a carrier, so here we have sickle cell anemia, and we also have what they call the sickle cell trait, meaning you have one copy of the bad gene versus having two copies of the bad gene. If we have two copies of the bad gene, all of our red blood cells are going to be funky. If we just have one, many of our red blood cells might be correct. So yeah, we would consider that similar. Yes. Go ahead.
So you just mentioned that we can turn off one of our genes. So a person with AS can turn off the S gene and just function on the A gene, which means they might not have like the sickle cell crisis often. But if they turn on the S gene, they might fall into that category of having the sickle cell crisis often. Is that what that means?
So it's not that we necessarily get to choose which one we turn on and off. The body decides that for the most part. And full disclosure, I don't fully understand the mechanisms of it, or I don't know them. But the body typically is able during the development process to figure out which genes are mutated versus which ones are not. So typically, if you're an individual who has the trait, you aren't, to begin with, you aren't going to have as many crises, just because at least some of your red blood cells are going to be correct. If you're someone who has two copies of the sickle cell gene that's mutated, you are pretty much guaranteed to exhibit sickle cell anemia. So in someone who carries the trait, I do think they still make some of the mutated hemoglobin because they don't always turn off that mutated gene. And it's actually, this is something that if you take biology for majors, I'm sure you'll really explore this. This is actually an evolutionary advantage to individuals living in Africa, because if you carry the sickle cell trait, some of your red blood cells, not all of them, but some of your red blood cells that are shaped like this, malaria, like Kira mentioned, the parasite that causes malaria can't live in a red blood cell that's shaped like this. So it's actually an advantage to have a mix of some of your cells that look like this and some of your cells that look like this if malaria is widely circulating in the population. Because someone who has all of their red blood cells looking like this are much more susceptible to having issues with malaria. So sickle cell trait can actually be a good thing if you are exposed to malaria. Sickle cell trait also less likely to give you the painful crises like Temiteo was talking about. Sickle cell anemia where all of your red blood cells are shaped like this, that's problematic. Even if you're around malaria, that's problematic because you're going to have all of those issues with blood cells getting caught in your blood vessels and causing issues. Yeah, so great questions about sickle cell. Absolutely. And a teaser for your introduction to biology class, because I'm very certain you will talk about that. All right. Well, I'm feeling the love. We've posed some good questions about lesson number 6. If there are no other pressing questions, I'm going to go ahead and move us into lesson number 7 to talk about the skin. So when we consider the skin, go back to our lovely skin classroom here. I saw some comments before. By the way, I didn't respond to them, but our evil looking sun over here. Yeah, I'm gonna show you that here on a slide soon. This evil looking sun is causing a couple of kinds of skin cancer. So he absolutely is evil, like we're seeing here. We're gonna talk about here in the skin lesson, we're gonna talk about the types of skin cancer. We're going to talk about the process of thermoregulation, so the way we be hot and cold, right? We'll talk a little bit about why tanning is bad. I'm going to get a little bit on my soapbox today. I'm going to be honest with you. I'm not sure if I've told you all this yet or not, but I worked for a dermatologist for a while. So, and she was a skin cancer dermatologist. So I'm going to get on my soapbox about tanning. But here's kind of an overview of where we're going today. We've got the lab exam coming up this week, right? We're all trying to forget that we have the lab exam coming up this week. But this is a good time for us to pause really quick and review some of our tissues. In the skin, we have 4 different tissue types. These 4 different tissue types, you're going to have to be able to identify them for the lab exam, but also for the next lecture exam. I'm going to do a little double dipping here. So we're going to see tissue images like this in lecture and in lab this go around. Let's start with my picture up here. Which kind of tissue am I looking at in this picture up here? What kind of tissue is this one here? Oh yeah, yep, this is adipose. Adipose, loose connective tissue. I'm going to abbreviate here. When we talk about adipose loose connective tissue, remember that this is one of those things I told you, let's double dip it. We need to know where it is in the skin and we need to know where it is in general. So when we're talking about adipose loose connective tissue in the skin, there's 2 names for the place where it's found. They mean the same thing. One of them is called the subcutaneous layer of the skin. That means exactly the same thing as the hypodermis. The hypodermis. So adipose loose connective tissue, this is the very bottom layer of the skin, the subcutaneous layer, also known as the hypodermis. We should know from unit number 1, what do I call those cells that live in adipose loose connective tissue? Yeah, this was actually a lecture thing, right? We call those cells adipocytes. Adipocytes. Remember that with adipocytes, they're these really large cells, and their entire space in here is filled with lipids. They're all about energy storage. And when we think about functions of this tissue, this is my energy storage, and this is my cushioning tissue. So adipose loose connective tissue found in the hypodermis, found in that lowest layer of the skin. Let's bounce to the opposite end of the spectrum here. This kind of tissue has a really long name. Do you want to type parts of its name for me? The one that I find in the epidermis, the outer layer of the skin. Yeah, so I'm seeing in my chat, let's have a little debate here. When we talk about the outermost layer of the skin, Keratinized or non-keratinized? The outermost layer of your skin. What do we think? Yeah, that outer layer of the skin is keratinized. Yep. Remember that keratin, and we'll talk about it more today, keratin is really tough. So this kind of tissue, which is, I'll type it here for us, keratinized, stratified, squamous epithelium filled with keratin. And that makes this kind of tissue really tough and resistant to damage. The outermost layer of your skin, we're got to be tough and resistant to damage, but the other thing that we've got to do too is got to have that UV protection. The UV protection comes from the type of cells we'll talk about in a moment called melanocytes. But most of the cells that I find in this part of the skin here, what's the name of most of the cells in the epidermis layer of the skin? Most of the cells in this tissue. Yeah, their name comes from the protein that they make, the keratin protein that they make. So most of the cells in the epidermis are called keratinocytes. Keratin builders, essentially, keratinocytes. So we've got the innermost layer of the skin that has adipose, loose connective tissue. We've got the outermost layer of the skin that has keratinized, stratified, squamous epithelium. So let's go to the middle then. We have which kind of tissue in this picture here? Which kind are we looking at here? Exactly. We're looking at areolar loose connective tissue. Areolar loose connective tissue is the kind of tissue that we find in an area called the papillary layer of the dermis. The papillary layer of the dermis. Areolar loose connective tissue has a whole lot of different protein fibers in it. See all of these thick red fibers and these little branching purple fibers? Because this kind of tissue has so many fibers in it, these little spots that I see in here, the cells in this tissue, I actually call these guys fiber builders, or my technical name for them is fibroblasts. So when we're in areolar loose connective tissue, and for that matter, any kind of connective tissue that has a ton of proteins in it, we always call those cells fibroblasts. When we think about areolar loose connective tissue, this is the kind of tissue that wraps around things or holds them together. The other place that we find it, besides the papillary layer of the dermis, is it's around a lot of our organs. So this is kind of a general connective tissue to connect things together, if you will. When I come over here to the other kind of tissue in the skin, this one has a whole lot more proteins in it. So this one is going to be an example of a dense connective tissue. Which particular kind of dense connective tissue is this hot mess over here? Yeah, exactly. This one is called dense irregular connective tissue. Dense irregular connective tissue. Getting its name because all of these proteins are irregularly arranged. Some of them are going parallel with the screen. Some of them are sticking out of the screen. Dense irregular connective tissue. This is the kind of connective tissue that we find in the place that's called the reticular layer of the dermis. The reticular layer of the dermis. And when we think about dense irregular connective tissue, because the proteins are arranged irregularly, this kind of tissue is really good at resisting pulling in multiple directions, because I have proteins that go all the different directions. Dense regular connective tissue, which we'll look at tomorrow in our histology review. Dense regular connective tissue is really good in one direction because all of its proteins are arranged the same way. But dense irregular connective tissue, that's good in all kinds of directions, like we see here. So when you think about the skin, kind of imagine the progression from outside to inside here. So we've got the epidermis with keratinized stratified squamous epithelium, the papillary layer of the dermis with the areolar loose connective tissue, the reticular layer of the dermis with dense irregular connective tissue, and then the hypodermis down below. So the hypodermis with that adipose tissue. You have this nice cartoon that makes the layers of the skin look so neat and organized. The big thing for us to keep in mind when we're thinking about the skin is we have these big regions in it. So make sure we know the 4 regions of the skin from the outermost layer, the epidermis, into the papillary layer of the dermis, which we're going to talk about these ridges called dermal papillae, down into the reticular layer of the dermis and that hypodermis down below it. We're going to start on the outside, the outermost layer of the skin. Remind me, when I talked about the outermost layer of the skin, we had stratified squamous epithelium. And what was the name of that protein that I had a ton of in that outermost layer of the skin? What's that protein I make in the outside? Yeah, exactly. The protein that I make in the outermost layer of my skin is keratin. Keratin. So this fills that outermost layer of the skin. And this is why we call it keratinized stratified squamous epithelium. So the outermost layer of the skin filled with cells called keratinocytes. They are keratinized. They're filled with keratin. When you think of the keratin protein, what I want you to think of is tough or strong. That's what the word keratin should make you think of. Let's look at what makes the keratin protein so tough and so strong. The keratin protein, when I look at its normal 3D shape, kind of looks like licorice. We got some more food analogies here. So here's my licorice of the keratin protein. By the way, looking at the keratin protein here, it's licorice shape. Which level of protein structure am I looking at when I look at the 3D shape of this keratin protein? What was the name for 3D shape again? Yeah, several of us have mentioned the overall 3D shape of a protein is called its tertiary structure. Tertiary structure, the overall 3D shape. When I look right here, I am seeing an alpha helix. And my alpha helix is one of my examples of which level of structure? My alpha helix. Alpha helix. Yeah, that's the one. Alpha helices are one of my secondary structures. Remember that the other secondary structure are those beta sheets. Beta sheets. When we talk about the secondary structure of keratin, the keratin protein has a ton of alpha helices. Lots of these curly Q ribbons. When I build these curly Q ribbons, though, notice how I make this stripe of hydrophobic amino acids. Hydrophobic amino acids. My favorite question today, what does this word hydrophobic mean? If these are hydrophobic amino acids, what does that mean? Yeah, they hate water, right? They don't like water, they're nonpolar. Okay, so your cells, as a friendly reminder, are filled with water. So I've got this protein that's got this big stripe of amino acids that all hate water. So this protein's pretty smart. Its secondary structure makes us have a straight line of hydrophobic amino acids. If I wrap this straight line of hydrophobic amino acids around another part of my keratin protein that also has a stripe of non-polar amino acids, I can squeeze together those non-polar parts. And when I squeeze those parts together, I push out the water. Again, here's that term that we just mentioned before, hydrophobic attractions, when we were talking about the hemoglobin protein. Part of what keeps these keratin proteins together is the fact that I've got hydrophobic parts that want to be right next to each other. So the keratin protein is a really strong protein because it's made of multiple parts of the amino acid chain that wrap around each other and squeeze together to push that water out. So keratin has the alpha helix secondary structure and then this licorice twisted up tertiary structure to keep the water away from my hydrophobic amino acids. With this really strong structure that our keratin proteins have, we can use them to build something called a desmosome. Now, a note I want to make sure you write down for yourself. Desmosomes are built with keratin proteins. Desmosomes are built with keratin proteins. So notice in my picture here, I've got a couple of cells that are next to each other. These cells have a place right here with a connection type called a desmosome. So desmosomes are a way for us to connect cells together that resist pulling in multiple directions. So you can pull me up, you can pull me down, you can pull all over the place. Desmosomes are going to hold our cells together. They resist pulling. The reason that desmosomes are really good at resisting pulling is because of all the keratin proteins that go into these junctions. I like to think about the keratin proteins that are found in desmosomes a lot like when you go into a store like Walmart. If you look up at the ceiling, how they have those metal rafters that are kind of holding the ceiling in place. Think about the keratin proteins kind of like that. Their job is to anchor these attachment sites called desmosomes in place in the cell. So I see some proteins where we reach from one cell to another to attach to each other. But what makes this junction, this desmosome, really strong is the fact that I've got keratin proteins pulling on it at all angles to keep it secure. So say this cell over here starts pulling this direction. On my other side of the junction, these proteins are like, oh, I got you. We're going to stay stuck together. Or if the desmosome starts pulling this direction, these proteins over here say, you're good. I got you. Or down here, if they're pulling this way, the proteins up here are going to be anchors to keep it together. So desmosomes, first thing we should always think when we think of this kind of junction, desmosomes have keratin. We use keratin proteins to build them. And then second thing we should be thinking is that they resist pulling. Desmosomes resist when we pull in various directions. So desmosomes, really strong junctions. The other way that I attach cells together that I find in the epidermis of the skin are what we call tight junctions. And just like their name tells us, tight junctions bring our cells together really tightly. We squeeze them together. I need you to write something down for me though. When we're talking about tight junctions, there is no keratin in tight junctions. No keratin used to build these tight junctions. If you look up close at them, so we can see them up here in our cells, they attach the membranes together of neighboring cells. So like their name suggests, it's a really tight junction. It's fusion. Nothing can get in between these types of junctions. But the proteins that I use to fuse the membranes together are not keratin. Keratin is only used in these desmosomes here. The goal of tight junctions is not to resist pulling in multiple directions like the desmosomes do. The goal of tight junctions is to make sure that no water can sneak in or sneak out. So when we're thinking about your skin, you are exposed to water every time you take a shower, every time we go swimming. It's almost summertime, right? So every time you go swimming or play in the sprinklers or wash the dishes, there's water on the outside of your body. We don't want all of that water to go straight inside your body. That'd be bad news. So we fuse together the plasma membranes of our keratinocytes using these tight junctions to make it so that water can't just freely go inside your body. Same thing with water leaving your body, right? When it's really hot and really dry around you, we don't want all your water to just leave the body. So tight junctions are something that helps to waterproof your skin. No water coming in, no water coming out. Tight junctions fuse the membranes together. Remind me, desmosomes, they don't fuse the membranes together, but what was their job? Why were desmosomes important? Yeah, exactly. Kaylin talks about kind of acting like an anchor or resisting pulling. Absolutely. So desmosomes really good at anchoring things together, helping them not pull apart. Tight junctions really good at sealing the membranes, waterproofing your skin. The other thing that helps to waterproof your skin are things called glycolipids. Glycolipids. Hey, their name tells us exactly what they are. Glyco means sugar. So glyco is sugar, like glucose, sugar. And lipids means lipids. This is a molecule that has sugar in one part and lipid tails in another part. Yeah, like Kira mentioned, literally my name means sugar fats. Sugar fats. So your skin makes these things called glycolipids, these sugar fats, and their job is to help with waterproofing as well. So Audrey mentioned in the chat asking if these are hydrophobic. What do we think? I'm going to put that question to the class. Hydrophobic? Yeah, there we go. Several of us are voting yes, and that is correct. Which part is it? If this is hydrophobic, is it the sugar or is it the lipids that make this hydrophobic? Which part's afraid of the water? Yeah, it's all about these guys down here. These glycolipids actually look a lot like the phospholipids, don't you think? We've got these two little lipid tails here, and then we've got this sugar head. Sugars tend to be polar, but lipids are always nonpolar. So glycolipids, sugar fats, these are hydrophobic as well. These do not like water. Now, the other name for glycolipids that will help us figure out where they are in the skin, they're sometimes called lamellar granules. Lamellar granules. I want you to make sure that you know that these two words mean the same thing. Glycolipids and lamellar granules, they mean the same thing. These lamellar granules, or these glycolipids, these are what cells make when they're in stratum granulosum. That's actually all these little purple dots that you see here in stratum granulosum. That's what those dots actually are. These little glycolipid molecules that the cell is making. See, I kind of like to think about glycolipids as the midlife crisis thing that keratinocytes do. So when we're down here in stratum basale, we're young, we're thriving, we're doing mitosis, we're making new cells. We grow up a little bit and we go here into stratum spinosum. By the way, stratum spinosum got its name because the cells have spines. Those spines are actually the desmosomes that we just talked about. So the spiny that you can kind of see, some of these attachments here, that's actually the desmosomes that we just discussed. So a cell starts in stratum basale. It does mitosis, gets pushed into stratum spinosum. And as other cells keep doing mitosis, it gets pushed all the way out here to stratum granulosum. And like I said, this is when the keratinocyte kind of goes through a midlife crisis. It says, oh no, I'm really far away from stratum basale. I've outgrown stratum spinosum. I am on my way to stratum corneum, where I'm going to die. I'm freaking out. And when I freak out, I make glycolipids. So in stratum granulosum, these cells start to make these little purple granules, lamellar granules, or glycolipids. They spit them out around them. When they spit out these lamellar granules, or these glycolipids, they automatically become waterproof. So when a cell covers itself in glycolipids, water can't get close to it anymore, which is great if you're in stratum granulosum or you're in stratum corneum, where there's water coming from the environment. It's not so great, though, when you're in the lower part here of stratum granulosum and your nutrients and your oxygen are trying to come this way from below. See, waterproofing knows no bounds. When I spit out glycolipids all around me, it's not like the water that has glucose in it is going to be less polar than regular water. When I cover myself in glycolipids, water from the outside can't get to me, but water from the inside can't get to me either. So in stratum granulosum, this is the layer where cells actually start to die. The reason that they're dying is because they're covering themselves in this waterproofing coat that keeps out the water from the environment, but also keeps out the water that has nutrients. So stratum granulosum, this is where the cells start to die. They're sacrificing themselves to make sure that no water gets into your skin, goes deeper, but it also cuts them off from their nutrients as well. So from stratum granulosum up, we're completely dead here in stratum corneum. We're dying here in stratum granulosum. Hey, if this was thick skin, which other stratum would I see as well? Which stratum is missing from this picture? Because this is thin skin. Yeah, exactly. Kara mentioned, I'm going to put in my little line here. The one that we are missing is the one called stratum lucidum, the clear layer, stratum lucidum. Excuse me, that layer would be here in between stratum granulosum and stratum corneum if we were in thick skin. Okay, so glycolipids and tight junctions. These are the two structures that waterproof the skin. Desmosomes and keratin, which is part of desmosomes, those are things that make the skin really tough. It's a good idea to try to group together some of these structures to help you remember how they work together, putting them in groups to remember some of these big ideas. Let's talk about another structure that helps to protect your skin. And the other structure that helps to protect your skin is melanin. For my friends who've had a chance to work on this lesson, what's the function of melanin? What does melanin do for our body? Melanin function. Yeah, so melanin is the specific protein that your body uses to absorb UV rays. Protein that absorbs UV. Oops. Let's pretend that that's a V and not a C. The protein that your body uses to absorb UV radiation. UV radiation, as we're going to talk about in a moment, has the ability to mutate your DNA, so to change your DNA. If it does that, if it changes your DNA, you can end up with, like we talked about with sickle cell, where we start to make the wrong protein, or where we make a protein that doesn't work anymore. It's particularly problematic if we mutate your DNA in a gene that helps to control your cell cycle. So if it's a gene that normally told your cell not to keep dividing, if we turn that gene off, if we mutate that gene, what will happen is your cell will keep dividing rapidly, and it will help to develop a tumor. That's what happens in cancer when our cells divide all the time. So the goal of melanin is essentially to prevent skin cancer. That's its goal. Melanin is made by these funky looking cells. Notice that they have these little extensions here called dendrites. We will use this word dendrites again when we talk about the nervous system. So the way that neurons receive messages is with their dendrites. The way that melanocytes share their melanin is by having these dendrites as well. Most of the cells, the vast majority of the cells in your epidermis, remember we called them keratinocytes. Keratinocytes are really good at making keratin, but they cannot make melanin. The only kind of cell that can make melanin is called a melanocyte. So melanocytes have these dendrites because that allows them to reach in between their neighboring cells and give them the melanin that they need to protect those cells, to protect themselves. Here's an important idea I want to make sure that you write down so you have this note. Everybody has the same number of melanocytes. Everybody has the same number of melanocytes. I'm kind of down here on this end of the spectrum. Do we have any redheads? Not sure if we have any redheads in our class. I don't have enough of our Bitmojis to know where we are on our spectrum here, but I'm kind of down here. Some of us probably are edging more toward the right side or more toward the left side of the skin color spectrum. All of us, though, regardless of what color you have, all of us have the same number of these cells. We all have the same number of melanocytes. Some of our melanocytes are more active than others. Do you want to take a guess? Is it more toward the left side or the right side of this spectrum where we see more melanin? More melanin. Yeah, the more melanin that you have, the darker your skin tone. The less melanin you have, the lighter your skin tone. And for that matter, also the lighter the color of your hair. And we actually even use melanin to dictate your eye color. So melanin is a protein that we see in a lot of different places. The cells that make melanin in the skin, the only cells that make melanin in the skin, are melanocytes. They make that melanin, they spit it out, these little melanin granules, they spit it out to cover all of the keratinocytes that live next door to them. The hope is by covering those keratinocytes in melanin, now the UV radiation won't reach their DNA and it won't mutate their DNA. So the job of a melanocyte is to make this protein that protects the DNA of your cells. Now, there is a way for you to increase the amount of melanin that your melanocytes make, and that's called tanning. So tanning is when you purposefully expose yourself to UV radiation, which is the kind of stuff that sun has, that has the ability to mutate your DNA. When you purposely expose yourself to that, your melanocytes say, oh my gosh, we're in trouble. There's UV radiation. We might get DNA mutated. So your melanocytes actually make more melanin when you're exposed to UV light. That's the physiology of getting a suntan, is making your melanocytes make more melanin because they were exposed to UV radiation. The problem with that is that melanocytes, while they're really good at making melanin and giving that melanin away to other types of cells, they're really bad at keeping enough melanin to protect themselves. So melanin or melanocytes are kind of like the people that you have in your life. Maybe you're one of them that's always taking care of other people and not really taking care of themselves. That's what happens with our melanocytes here. So you go tanning and you're exposed to UV radiation. Melanocytes make a whole bunch more melanin, so much melanin they're crying about it. They're giving that melanin away to your other cells. They're not keeping it for themselves. And the UV radiation can actually come into the cell and mutate the melanocytes' DNA. Anyone want to help me? What would it be called when a melanocyte gets mutated? What would I call that if my melanocytes' DNA gets mutated and it becomes a cancerous cell? Yeah, Kira's right. That's what causes melanoma. Melanoma is when a melanocyte, my melanin producing cell, oh, Kaylin says I'm cutting out. Melanoma is when those melanocytes become cancerous, when they replicate themselves. This is why when someone has melanoma, their cancerous lesion is highly colored. So a melanoma, you're going to see a dark brown color. You might see a purple or a red. A skin cancer that's caused by an issue with the melanocytes, we're going to have extra melanin in that skin cancer. That's what we see here. Sometimes it's not the melanocytes that become cancerous. Sometimes what becomes cancerous are keratinocytes. Keratinocytes are the cells that make up most of the epidermis, right? So the keratinocytes. Melanocytes sprinkled in here and there in various little places, but most of our cells, keratinocytes. If one of your keratinocytes that lives next door to the melanocytes becomes cancerous, so if a keratinocyte in stratum basal becomes cancerous, stratum basal, if one of those keratinocytes becomes cancerous, you develop what's called a basal cell carcinoma. A basal cell carcinoma. Basal cell carcinoma is the most common kind of skin cancer. It's also a more slow-growing form of skin cancer. The good thing about basal cell carcinoma is notice how our cancerous lesions, so my cancer cells, all kind of grow down together in a mass. This means it's going to be easier for your dermatologist to remove all of those cancer cells. So when I worked for a dermatologist, we used a procedure called Mohs Surgery, where we would literally go through and we would cut out a piece of tissue and look at it under a microscope to see, oh, there's still cancerous cells at the bottom. Let's go through and cut some more tissue out. And we check, still cancer cells. You keep cutting little pieces of tissue until you cut out a piece of tissue and there's no more cancer in it. So for a basal cell carcinoma, because the cells all grow down together, it's going to be easier to remove the entire lesion. Basal cell carcinoma, most common, least deadly. Squamous cell carcinoma is what happens when cells that live in stratum spinosum become cancerous. Stratum spinosum become cancerous. When these become cancerous, they grow a little bit more quickly, and they also don't necessarily grow in a group like we see here. See how there's kind of this random line of cells that are starting to grow down? So it's going to be harder for a dermatologist to get all of the cancer cells, because if they miss the fact that there was still this little line of cells down here, those cells might keep growing. So squamous cell carcinoma is a little bit harder to get fully excised. They grow a little bit faster than the basal cell carcinomas grow. The most serious, though, the fastest growing, is melanoma. And we already talked about this. When we're talking about melanoma, which kind of cells became cancerous again? How did this cancer start? Who's the cancer cell here? Yeah, so when we talk about a melanoma, the cell that became cancerous were these melanocytes, the melanin producing cells. That's why, again, when you look at these lesions, these have a lot of color, melanin production. I'll flash this up really fast. I can't remember, did I include this in the guided lesson or not? I think I may not have. Oh, I did. Okay, perfect. So just a fun cartoon for you to show you some of the differences between basal cell carcinoma and squamous cell carcinoma. Both of these kinds of skin cancers develop when keratinocytes, the normal cells of the epidermis, when they become cancerous. So two types of cancer that form from the keratinocytes, and then the kind of cancer that develops because of melanocytes called melanoma. When you're thinking about melanoma, melanoma is often or is always diagnosed in some stage. So some stage of cancer, cancer is always staged. Particularly in melanoma, though, we can relate this to some of the anatomy that we've learned of the skin, how we determine which stage the melanoma is. Best case scenario for catching melanoma, if you're a patient that has melanoma, is to catch it when it's stage 0. Stage 0 melanoma, notice that this is when the melanoma is all in the epidermis. It's completely confined to the epidermis. So stage 0, there's no spread. There's no, here's the word for spread, metastasis. There's no metastasis, no spreading of the cancer. In stage 1 melanoma, we've started to get into the first layer that's right underneath the epidermis. So in stage 1 melanoma, these melanocytes not only are found in the epidermis, now they're into the papillary layer of the dermis. In stage 2 melanoma, we're into the reticular layer of the dermis. By stage 3, we've reached the hypodermis, and inside the hypodermis, in that adipose tissue, we also have things called lymph nodes. And lymph nodes are those things in your neck that the doctor feels when you're sick. Lymph nodes are the place where your body typically does immune reactions. So when a melanoma reaches your lymph nodes, your immune system might recognize that it's bad, or it might not. But the problem with these melanocytes getting to the lymph nodes is the lymph nodes and the lymphatic system is a super highway throughout your body. So it's typically a bad marker for any kind of cancer if they find cancer cells in the lymph nodes that are near that region. Because if the cancer cells have reached the nearby lymph nodes, there's a very good chance that they've found other places in the body that they've also spread to. So stage 4 melanoma, like stage 4 with any kind of cancer, we would see those metastases, the mets, if you will. So it metastasizes to other places in the body. We start to develop tumors in other parts of the skin where maybe there wasn't the UV exposure, but the melanocytes traveled there through the body. Or we see the metastases in the liver or in the lungs. So stage 4 melanoma is when we've spread to other organs. Stage 3 is when we found that superhighway. Stage 2 and below is all confined to the skin. So the stages of melanoma, this is something that we do need to be familiar with. Yeah, Kaylin said it's very similar to the burns. Absolutely. Yeah, so it's the same kind of idea. Which part of the skin or, beyond the skin are we found in? That's what we're seeing here with our stages of melanoma. A big difference between stage 0 melanoma and stage 1 melanoma is whether or not we've left the epidermis to go into the dermis layer. Now that we have talked about all the ideas of the epidermis, let's go into a discussion here of some of the characteristics of the dermis found underneath it. When we talk about the dermis, there's a structure that we mentioned in lab briefly, but now we'll talk about it more in person. There's these things called the dermal papillae. So dermal papillae, this papilla word kind of means fingers. When you think about the dermis layer and the epidermis layer coming together to form your skin, they don't actually meet in a straight line. They meet in this up and down pattern, like we can see here, the up and down wavy pattern. So this wavy pattern is the dermal papillae, the fingers of the dermis, and the epidermal ridges, where the epidermis meets the dermal papillae. These make what we call the friction ridges, So the surface of your skin, rather, what I don't like about this picture is it's flat up there. You would normally see a pattern on the surface of the skin that matches those dermal papillae. So we'd have ridges or raised parts of the epidermis that form because of the dermal papillae. Those ridges are called friction ridges, or in easy person words, they're called your fingerprints or your toe prints. So there's 3 different patterns that we can see in our fingerprints. It's this arch pattern where we go up and down, kind of arch around it. There's the loop pattern where there's a specific place that everything loops around. And then there's that the squiggly whirl pattern that you see there. At least half of you are staring at your fingers. It's totally fine. I get it. Each of your fingertips are going to have a slightly different pattern to them. And every single person actually has different fingerprints. Even identical twins have different fingerprints. The reason for that is because everyone's dermal papillae develop differently during the embryonic development process. So it's very early on in development that we start to see these fingerprints. But again, the fingerprints are unique to every person. Even identical twins that have the same DNA, their fingerprints won't be identical to each other. So fingerprints form from the interaction of the dermis layer of the skin with the epidermis layer of the skin. That, by the way, is why your fingerprints are permanent, because the way that your dermis interacts with your epidermis is the same your entire life. We never redo that. We never change that on you. I am about out of time, so I don't think I want to dive into thermoregulation. So I don't think I want to start that and not have enough time to complete it. So we will plan to finish up lesson number 7 next week, and we'll work through lesson number 8 next week as well. Yeah, I see the notes in the chat. We can't type and look at our fingers at the same time. Yeah, that is very true. Yep. So I'm going to call that a day for our content. Let me see if I have my updated schedule for us. Yeah, so the lesson number 7 homework, you still have plenty of time on that. The lesson number 6 stuff, that's due on Friday. So make sure we're getting our transcription and translation, the Central Dogma stuff. That needs to be done by Friday. This lesson, we've got plenty of time. It's not due till the next week. So we'll make sure to wrap it up early next week and then dive into lesson number 8. Any last minute questions before I end our recording? Here comes our daily penguin. Daily penguin loves the skin. Yeah, so Audrey mentioned that we're having a lot of Friday due dates. Yeah, that's because I switched some stuff to give you some extra time. So today, what's today, Tuesday? Yeah, there's nothing due today. This Friday of this week, make sure you've wrapped up lesson number 6, the Central Dogma stuff. Next Friday, so the Friday right before spring break, is when the stuff about the skin will be due. My wink wink nudge nudge do on that though is as soon as we finish covering lesson number 7 together, I highly suggest that you start working on the homework assignment. Don't put it off until Friday because we're going to move straight on into lesson number 8 and do lesson number 8 stuff. So plan to get lesson number 7 done ASAP too, but the thing you have to get done by this Friday is number 6. The lab exam opens up Friday morning and it closes Sunday night. So you've got 48 hours to find the best time to take it in. I'd recommend let's get lesson number 6 homework assignment done like tonight or tomorrow, because then you're not worrying about lecture stuff at all. You're just focusing on the lab exam stuff. Yeah, the penguin's waving goodbye. So long, Mr. Penguin. He'll come back again tomorrow. All right, well, I'm going to go ahead and stop the recording. I will stick around if we have any last minute questions. Otherwise, remember that tomorrow is a histology review. So bring your microscope eyes, be ready for lots and lots of tissues tomorrow. And on Friday, we will do a general midterm review. So have a great afternoon, and I hope to see most of you back again tomorrow.